Today, I will describe Cumulus. This is a cloud-based single-cell and single-nucleus genomics analysis framework my lab has developed in the past 2 years. I will start my talk with motivation and then introduce the Cumulus system. Next, I will benchmark Cumulus with alternatives on large data sets and demonstrate Cumulus interactive visualization and analysis capabilities, which are super useful. And then I will show you how awesome our Cumulus team is. We keep developing new features rapidly. And in particular, I will discuss 3 recently updates in details. Last but not least, I will discuss the Cumulus Roadmap, what we plan to do in the next 5 years. So we all know that human immune system is super important because immune cells are actually presented in all human tissues, and they are likely to be involved in all diseases. And actually, they are next generation therapeutic targets for a wide range of diseases, for example, cancer immunotherapy. Thus, at the Broad Institute, we have initialized several immune cell atlas efforts. These include the efforts of human hematopoiesis, as well as across humanized mouse and different organs. As a computational lead, I'm in charge of the atlases of human hematopoiesis system, which includes cells from bone marrow, cord blood, and peripheral blood. For this data set, we have profiled around 1.5 million single cells using single-cell RNA-seq to be representative of the human immune system. For each tissue type, we actually collected samples from multiple donors with equal gender representation. This is so far still the largest single-cell transcriptomics data in the world for the human immune system. Although, 2 years earlier, I'll claim this is the largest single-cell data set in the world, but now it's not, but still is the largest transcriptomic data, single-cell transcriptomic data for the human immune system in particular. So for 1.5 million single cells, we obtained over 5 terabytes of sequencing data. Although like for big tech like Google and Facebook, 5 terabytes is not that big, but for biologists, this is really a gigantic number. And our data actually posed serious problems for the analysis. There are 2 problems. First, computing resources are not enough. And second, most existing tools, at least at that time, could not scale up. Therefore, together with my team, including Yimin Yang and Josh Gott, we developed Cumulus. This is the first comprehensive cloud-based framework to address the above 2 problems. Before I introduce Cumulus, let me first briefly describe the workflow of single-cell RNA-seq data analysis. We first prepare single-cell CDN libraries from either droplet-based or plate-based protocols. We then send these libraries for sequencing. And after sequencing, we start the analysis flow. We first trigger this is called the make-fast-q step. This step extracts raw sequencing reads from the sequencer. The next step is a count step. And in this step, we align the reads to a genome and generate this gene by count matrix for each sample. Lastly, we trigger the analysis step to discover new biology. This step pulls gene count matrices from all samples, finds cell clusters based on expression profiles, annotate these clusters with putative cell types, and visualize them in, say, for example, 2D space. So the make-fast-q and count steps, they are easily parallelizable if we have enough computing resources. Thus, we implemented Cumulus based on Broad Institute's Terra platform and the underlying Google Cloud Platform, so that Cumulus can utilize hundreds of thousands of CPUs and now also GPUs on cloud to run these two steps in parallel. And in particular, for 10x genomics data, we have a cloud-based wrapper of Cellranger for the count step. So because I mentioned like Cumulus is built on Broad Institute's Terra platform and also Google Cloud platform, you may ask, if I can use it, say, for example, in a local big cluster or other alternative cloud providers. So here I want to say, although Cumulus is built on top of Terra and Google Cloud, it is readily adapted to alternative platforms because Cumulus only depends on Docker images and workflow description language files. And Cumulus can run on any backend Cromwell. This is a Widow execution engine Cromwell supports. This includes Amazon Web Services, Google Cloud Platform, local computing servers, and a variety of computing cluster backends. And we can make running Cumulus jobs even easier by using Kepper. This is a tool developed by Encode DCC. And it's a Python command line tool. It's a Python command line wrapper for the Cromwell. And this will make, a command line. This will make running like jobs in command line much easier. However, I have to warn you, like for alternative platforms, we only have these command line tools provided, which makes it a challenge for biologists without a programming background to run Cumulus. So for the next step, the analysis step, this step is much harder to parallelize across different servers. Therefore, we developed an ultra-fast Python analysis module. We call it Pegasus, which can run on cloud or local computer. Pegasus covers most common analysis tasks. It can filter low-quality cells and genes, select highly variable genes, perform batch correction, conduct PCA, build nearest neighbor graph, and find clusters using community detection measures such as Lewin and Leiden algorithms, and visualize data using either t-SNE or UMAP. For developmental data, we can estimate diffusion maps and visualize the data in force direct layout embeddings. Lastly, this Pegasus package can perform differential expression analysis, detect cluster-specific markers, and annotate putative cell types based on known markers. We have actually made several algorithmic and implementation improvements in order to make Pegasus scalable. And here is one of the improvements we have made. I just want to show as a demonstration. This technique, in theory, can speed up the calculation of any two-dimensional visualization. And the motivation is, we know that large data sets are often redundant. Therefore, we might be able to capture the global structure using only a fraction of the cells to speed up the visualization. Thus, we propose the following algorithm. The first step, we downsample a subset of cells inversely proportional to each cell's local density. This is measured by its median k-nearest neighbor distance. And the goal here is we can, with this like non-uniform sampling, we can sample more cells in the sparse regions, which might represent rare cell types, and sample less frequent from the dense regions, which most of cells may like represent the same cell type. And then the second step, we project the sampled cells using an embedding algorithm of your choice. For example, you can do t-SNE, you can do UMAP, you can do force direct layout. And then we train a deep learning regressor using the embedded coordinates as a ground truth. Once we have this regressor, instead of running the embedding algorithm that can be time consuming, we directly predict the coordinates using this regressor for all other cells. And actually this idea is inspired by Cho et al. Cell systems paper. And lastly, because in most of cases, this regressor does an okay job for prediction for the coordinate prediction, but not 100% accurate. We need a last adjustment step. In this step, we basically adjust the predicted coordinates by running the same embedding algorithm for only a small number of iterations. And let's see this algorithm in action for speeding up the UMAP. And we call the resulting visualization as Net-UMAP. So as a first step, we generate a UMAP by, say, subsampling 10% of the cells. Here different colors represent different cell types annotated separately using known markers. And then using these UMAP coordinates, we can train a regressor and use this regressor to predict the other 90% of the cell. And here you can see this UMAP actually is blurred. And in the last step, we just ran the UMAP for, say, 10 iterations to make the UMAP crystal clear again. And our algorithm actually can speed up the visualization of UMAP, like t-SNE, and force direct layout by more than twofold change, by more than two-folds, while keep the quality roughly the same. So by the way, any questions so far about this algorithm or like other things before we move to the benchmarking section? If there's no question, I'll just move on.
Sir, I have a quick question.
Yep, sure.
Have you basically done a similar analysis, but with, like, let's say an organoid where you kind of have a constrained set of cells and you have sort of intercellular sort of interactions that might actually perturb their kind of representation within your visualization here?
Sorry, can you repeat your question again? Why is organoids like play a factor here?
Well, that's precisely a question that I'm interested in kind of looking at myself. So I'm curious if basically you had a multidimensional sort of tissue structure, if you would actually see a difference in sort of how these cells end up separating on these signals. Maybe this is I might be misunderstanding something. Maybe this is a better question for later.
Okay. So this algorithm, the basic is like in big data field, there's all these kind of sketching algorithm. But all the assumption is because once you have large enough data points, by like non-uniform or uniform sampling of this data point, you can capture the global structure without lose much accuracy. And then like you just sampling a small subset of cells, build a skeleton of your visualization, and then basically you fill in the other data sets to make the details much clearer. That's the concept.
Ah, got it. Thank you.
Mhm.
Can I ask a question quickly as well?
Yeah, sure, of course.
Does this Net-UMAP or Net-TSNE have similar performance characteristics in 3D versus 2D projections?
That's actually a great question. We have never tested in 3D. But I suspect we should do a similar good job. Yeah, but I never tested it.
Cool, thank you.
Mm-hmm. Okay, so in the benchmark section, we will first benchmark Pegasus, which is a Python analysis module with Seurat and ScanPy. And these are basically because Pegasus perform similar functionalities than this to these Seurat and ScanPy packages. And we'll do the benchmark on two data sets. One is bone marrow data sets from our Immune Cell Atlas project. This data set consists of around 300,000 cells. The other data set is 10x genomics, 1.3 million mouse brain mouse brain data sets. This is just used to test the limits of each software. And after that, we will benchmark Cumulus. This is our cloud solution with a traditional single server solution that runs Cellranger plus either Seurat or ScanPy on the bone marrow datasets. And here I listed the software versions used for benchmarking. So for bone marrow dataset, we benchmarked 10 key analysis tasks using a single server with 28 threads and 256 gigabytes of memory. And we highlighted the best performer for each task in bold. And here you can see that Pegasus performed best for all 10 tasks. And we have similar results for the 1.3 million mouse brain cell dataset. Note that Seurat even failed to load the whole data matrix into memory, and this is omitted in this comparison. And another note is here, basically, I just show you the speed of Pegasus is really awesome. Maybe you have concerns about if they produce results of comparable quality or not. I did not include slides here, but if you're interested, feel free to find the comparison in my Cumulus paper. This is a Nature Method paper, and you can find the results in supplementary figures. So, and this table shows that the analysis supported by each package are actually similar, although each one has its unique features. And the red check marks are actually analysis we recently supported. And this table compared running Cumulus on cloud with a single server solution. As you can see here, Cumulus successfully reduced the analysis time from 10 to 9 days to 15 hours. And in addition, the computational cost is modest. For analyzing one sample of around 4,000 single cells, Cumulus costed less than $2, while the experimental cost can be as high as 1,500. Besides fast and cost-effective, Cumulus also offers a comprehensive set of features. It processes both droplet-based and place-based data. In addition, it supports site-seq, which measures the transcriptome and epitope abundances simultaneously, and PerturbSeq, which is designed for pooled CRISPR screens. Cumulus also supports a variety of sample pooling and demultiplexing strategies. These include pooling by donor-specific genetic information, cell hashing, and nucleus hashing. And then you may ask, great, Cumulus basically for the concept runs Cellranger. But we also recently have this development of new count matrix generation tools, such as Human Cell Atlas Optimus workflow. This is cloud-based workflow for generating the count matrix, and all these Alivin. This is Salmon team developed this for single cell RNA-seq, and Callisto bus tools, and Star Solo. So you may wonder, how are these new tools compared with the Cellranger? So to answer this question, we did this benchmarking. We basically run a 5,000 PBMC dataset. This is 10x genomics V3 chemistry, publicly available. And this dataset consists of around 400 million reads. And we benchmark each of the two on cloud for their execution time and cost. And we give them 32 CPU. We give each tool a Google Cloud virtual machine of 33 CPUs, 120 gigabytes of memory, 500 gigabytes of disk space. And here is the results. You can see that both the new tools, Alivin, Callisto bus tools, and Star Solo, they run much faster than Cellranger. And as a result of spending less time, they also cost less. However, we can see the best performer actually are the Star Solo and Callisto bus tools. They all finish within 1 hour, and the cost is less than 30 cents. But there's one caveat here. For Callisto bus tools, this aligns to the transcriptome reference instead of the genome reference. But the Star Solo aligns to the genome directly. Any questions so far? Okay, so the next section, I want to discuss about Cumulus interactive data visualization and analysis capabilities. Once users finish their Cumulus jobs, they can instantly visualize the results interactively using Cirrocumulus. This is a cloud-based serverless visualizer we have developed here. Alternatively, they can download the analysis results, say in H5AD format, locally, and visualize them using third-party tools, such as the CellXGene or UCSC Cell Browser. And if you're using the Terra platform, you can also conduct post-hoc analysis interactively using Terra notebooks without the need of downloading any data objects locally. Lastly, once you finish your analysis, you can deposit your results to the single cell portal seamlessly for publication purpose, because Cumulus actually generates single cell portal compatible files. And here is a demo of the Cirrocumulus visualizer in action. To import the data, you just need to provide the dataset name and the link and the result location on cloud. You can share your data by typing with your collaborators by typing their email address. And then you click Save. If you load in the data, using Cumulus, you can visualize either categorical features like the cluster, like the cell types shown here, or continuous types such as gene expressions shown here. You can also select a subset of cells and observe it across multiple features, as demonstrated here. And you can also visualize a dot plot interactively. Lastly, if you want to share some visualization results with your collaborators, you can easily click the copy link button and send them with an email of their link. Cirrocumulus now also supports spatial transcriptomics data visualization. And here is a demo of the 10x wisdom data. Each dot here is colored by the cluster it belongs to. And Terra users can interactively analyze their data using Pegasus on cloud. Here is an example of generating beautiful heat maps and dot plots using Pegasus. And for more examples, you can visit the Cumulus featured workspace listed here. And here is a summary of the Cumulus highlights. Cumulus paper is published in Nature Methods. We already have a large user base. The Python package has already over 22,000 downloads. And major cell atlas projects, and there are several major cell atlas projects that either used or decided to use Cumulus. These include the Human Immune Cell Atlas, the Human Tumor Atlas Pilot Project, the Human Cell Atlas Data Coordination Platform, Internal Investigation, and the LongMap 2. And there are other consortiums that are actively evaluating Cumulus for their use. These include the Human Tumor Atlas Network funded by NCI and the Brain Initiative Cell Census Network. So here, I just want to say our team is really excellent at rapid development of new features. And here I'll show you the Cumulus development timeline since its paper submission. We released a PyTorch implementation of the Harmony data integration algorithm in January. We added a new algorithm to calculate gene module score in May. We deployed a customized Cumulus for the Penn Boston COVID-19 autopsy and blood research efforts in early June, and incorporate another data integration tool, Scanorama, in late June. And our paper is published in late July. And we basically rewrote our plotting library so that we can generate all these nice figures for publication in early August. And we speed up the differential expression analysis by 26-fold change in September, and provided preliminary nanostring spatial transcriptomic data support. This is in late October. This is not publicly available yet. However, and we actually work closely with the users who help us to gear Cumulus towards the right direction. And in the following, I'll discuss 3 of these recent updates. The Harmony-PyTorch implementation, module score calculation and differential expression analysis speed up. So Harmony-PyTorch is a PyTorch implementation of the Harmony algorithm published in Nature Methods. And the original implementation is written in R. And the reason we do this PyTorch re-implementation is PyTorch, this re-implementation actually is more scalable and allows us to do computation on GPUs. And here I will benchmark the Harmony-PyTorch on 3 data sets. One is small, one is medium-sized, and one is large. First, this plot shows the Pearson correlations between batch-corrected principal components computed using the original Harmony and Harmony-PyTorch. And this figure just suggests Harmony-PyTorch faithfully recapitulates the original Harmony algorithm. And this table shows Harmony PyTorch is more scalable on large data sets. And in particular, for the large data set, we can see the CPU version is 7.5 times faster, and the GPU version is 14.6 times faster. The error bars here were calculated based on 10 independent runs. The next thing is we want to discuss how to calculate gene module scores. Given a list of genes describing, say, one gene module or one pathway, biologists always want us to calculate summarized score so that they can know which cells are expressing this gene module or not. And in addition, when we calculate this gene module score, we also need to remove technical noise related to gene abundance. And here, I show the cell type annotated UMAP of our bone marrow dataset on the left. And on the right side, I just show the B cell module score based on these 10 B cell markers. And here we can observe that the B cell scores are high in B cells and low in all other cells, which means this is a like working module score. The traditional methods for calculating gene module score, as ones used in both Seurat and SciMPy, they approximate the module score by sampling. And instead, we found a closed form solution that can compute the exact module score. And as shown here, for the B cell module score on our bone marrow dataset, we show on the y-axis the Spearman rank correlation between SciMPy and Seurat's module scores at different sampling size with our exact module scores. And we can observe that both SciMPy and Seurat scores approached our scores when the sampling size is increasing. In addition, it seems Seurat's module score is better than SciMPy's because Seurat's curve dominates SciMPy's curve. Note that the sampling size actually stops at 500 for Seurat because Seurat basically crashed on sampling size above 500. Besides accurate, our calculation is also 20 times and 8 times faster than the Seurat and SciMPy computation thanks to like the exact formula we found. Our last improvement is the speed up of differential expression analysis. Inspired by Presto, we redesigned our differential expression module, which significantly speed up the Mann-Whitney U-test and area under ROC curve calculation. This MW test is also called Wilcoxon rank sum test. It is a non-parametric test that is more robust and thus preferable than the t-test. And these AUROC values is also very useful based on feedbacks from our biology collaborators for select cluster-specific gene markers. And we tested our redesigned module on a subset of bone marrow cells consisting of 35,000 cells. And we achieve a 6.2 times speed up compared to our original implementation. And because it's speed up, now Pegasus calculate differential expression using this MW test and calculate AUROC values for all genes. And if you are familiar with either Serrat or SCAMPy, you know that they can also calculate differential expression for all genes, but by default, they only calculate differential expression for highly variable genes, basically because of the computational bottleneck. But because they speed up, basically we can do all differential expression gene for all the analysis for all the genes. So any questions so far before I go to the last section of Cumulus Roadmap? Okay. So here is what we plan to do for the Cumulus. We plan to extend Cumulus for single-cell multimodal omics. This includes support of large-scale data analysis of single-cell ATAC-seq, single-cell immune raptor sequencing, spatial transcriptomics and proteomics, mass cytometry, and also support multimodal analysis. This means joint analysis of single-cell ATAC-seq, RNA expression, and maybe site-seq for protein expression. So we'll discuss each aspect separately. For single-cell ATAC-seq, currently Cumulus supports Cellranger ATAC-seq, Cellranger attack on cloud. However, in the future, we plan to extend Pegasus for single-cell ATAC-seq downstream analysis. And in particular, we want to address the big data challenge for the single-cell ATAC-seq field. As we know, the feature size here for single-cell ATAC-seq, each feature is not a gene's expression, but a separate peak or a separate genomic bins. The feature size for single-cell ATAC-seq is 10 to 20 times larger than the size of single-cell RNA-seq. And this will cause big troubles for both generating the feature by cell matrix and downstream analysis. For generating the feature by cell matrix, I will collaborate with Dr. Heng Li's lab on incorporating their newly developed tool called ChromeMap, which can speed up the computation by 60 times compared to Cellranger attack. And for the downstream analysis, we often need to actually pull matrices from multiple samples together. And then it is possible we even cannot load the full data set into memory. Thus, we will design novel online learning algorithm to address this memory challenge. Josh also have these similar online learning algorithms for his awesome Liger tool, online non-active matrix factorization. And the good thing about this online algorithm is you do not need to load all the data into the memory. And it also enjoys a very fast convergence rate. For the single-cell immunoraptor analysis, currently Cumulus supports Cellranger VDJ on cloud, and our planned development include evaluating alternative VDJ assemblers, such as Trust4 developed by Shirley Liu's lab. This is very important because actually Cellranger has its own VDJ assembler that is never peer-reviewed or published. And actually, between their versions, say Cellranger 3 and Cellranger 4, and now they have Cellranger 5, they keep changing their algorithm. So, for users, it's just like a black box. You do not know, what they give you as output. So it's very important we can have a systematic benchmark of this tool and other available tools. And we'll also try to extend Pegasus to support more downstream analysis. For example, based on both single-cell RNA-seq and the T-cell receptors, we can detect clonal types in single-cell clusters. Our next direction is spatial data analysis. For spatial data analysis, this is still in its infancy. For here, I'm aiming at developing a standard operation procedure for spatial analysis. For example, ideally, users only need to type some inputs, tell the machine where the data is, and click the run analysis button. Then for different type of the spatial data, for example, include slide-seq, 10x WISM, nanostring GeoMX, and merfish data, it will trigger different data processing workflows for different data type, and then generate a unified format data for the gene expression and spatial information. And then for this unified format data, we can put them into Pegasus to do analysis such as clustering, differential expression analysis. This actually shared with single-cell RNA-seq data analysis, or like spatial data specific analysis, like cell type deconvolution and detecting spatially variable genes. And last but not least, we also need an efficient data structure for organizing single-cell multimodal omics data. And data is a very good example for efficient data structure, but this data structure is only designed for one modality. For example, if you have both single-cell RNA-seq and single-cell TAC-seq, you need to store them in two data object, which is not a very clean solution. Therefore, we developed what called a new data structure called Pegasus IO. And in this data structure, users access a multi-model data object, which can contain a list of unimodal data objects. And the unimodal data object basically resembles AND data, but it can specialize to a variety of modalities, such as ATAC-seq, VDJ, mass cytometry, site-seq, or nanostring data. And for example, and then each of these specialization have like functionalities to process the specific data type. For example, for the cytometry data, it has ability to are processing my cytometry data in a specific format they call flow cytometry standard format. Yeah. And last, I want to say both Cumulus and Bro's Terra platform are free to public, but you still need to pay pay Google Cloud for computation storage. And here we have the and Cumulus is open source and the BSD3 license, and the Pegasus actually is a Python package you can run on your laptop. If you're interested, please give it a try. And we also have this Harmony-PyTorch as a separate Python package for you. In the end, I'd like to thank for my colleagues and collaborators. And in particular, I'd like to thank the Cumulus team, Yiming Yang and Josh God, and my collaborators at the RegApp Lab, Villani Lab, and Hakohen Lab. And thank you. Now I'm good for questions.
Okay, so thank you both for this wonderful talk. So do we have any questions from the audience? Please feel free to unmute yourself and ask the question directly to Bo.
Well, that was a great talk. Thanks.
Thank you.
I wanted to ask about the PyTorch Harmony implementation. Did you change the algorithm at all? Like, for example, converting it to a mini batch optimization instead of using all of the data points at once, or was it simply just rewriting it in the PyTorch?
It's just a simple rewrite in PyTorch. Yeah.
I see.
Yeah.
That's interesting. I was curious why just rewriting it in PyTorch gave such a big speed up.
Oh, let me share the screen. Harmony-PyTorch. Yeah. So there's a couple of reasons. I think one of the reasons is the original R implementation, although for say the time-consuming matrix multiplication, it's implemented in C or C++, but I think it does not utilize multi-thread very well. However, as part of the PyTorch implementation, PyTorch basically depends on this torch implementation. They did specific optimization for matrix multiplication and can automatically do parallel computation when do this matrix operations. And I think this is a reason why the CPU implementation is much faster. Actually, we also have a Python implementation using NumPy and just using NumPy. And that version, if you benchmark it with Harmony R, they basically perform the same. You do not see a big speed up.
Got it. So are you using multiple cores on your CPU for this comparison?
Oh, yes.
I see. Okay, so it's really the multi-core that makes the difference.
Yes, the multi-core, so if we do single core, it's still faster, but for this dramatic speed up, yes, it's a multi-core. I think we benchmark it on a 20-core CPU, server, something like that, yeah.
Okay, great, thanks.
Mm-hmm.
So, Bo, I have another question. I think it's really great. Many of these single cell consortium use Cumulus to host their data. So I wonder if I want to use this consortium data like Human Cell Atlas data, can I just use the data from your platform, or have to download somewhere to like a bucket and run it myself? So can you give me some like instruction on how to use?
Oh, is that like you want to reanalyze the data, or? Yeah. Say, it's a bit complicated. For example, the Human Cell Atlas, I think they hosted their data in a website created by Sanger Institute in UK. Okay. And then you can download these spam files or these count matrices directly. And I believe you can also import the data to the Terra platform directly.
I see. Great.
Yeah. And if you do the Terra route, then you can run like all these workflows like available in this environment. Yeah.
And another question is, if I want to, for example, I want to like build a GAN, like adversarial, like generative model over this large scale, like cell cohorts. So can I still use your platform to do that? Or I have to do like this type of GAN learning or generative model, can I write on your platform or not?
Oh, Cumulus basically is a way we try to standardize single cell genomics analysis for biologists. But actually it's several layers. So for all these kind of things, the bottom layer actually is Google Cloud Platform. And this is basically the same as you use either Amazon or Microsoft. It's just cloud platform. You just pay it and you use it. And then what Terra is actually is a thin application layer that handles a workflow execution and for you and give you a very nice user interface. For example, like say, if for some reason one of your cloud nodes crashed, and you want the ability of automatically retry it, or running it from the cache results, Terra can help you with you. And then for you say, if you want to do a GAN or whatever, you can just write your own tools and import it to Terra and to analyze all of your data. And then like Cumulus basically is a set of standard tools like we set up to like make analysis more automatic.
I see. So basically, I can still write new deep learning model on.
Of course.
Okay, I see.
Yeah. And I think your lab also have like GPU servers, right?
Right.
Yeah. So in that case, like for just benchmarking or testing, you can just run on your own server. But say if you really need a large computation, you can just do these cloud things, and you can ask unlimited GPUs. But I have to warn you, it costs a lot.
Okay, I see. And so far you haven't worked on like single cell high-C data for the like data frame or the object, right?
Not yet, but I'm actually very interested in like working on this. Maybe we can talk more about it if you have needs and whatever.
That's great. Okay, great. Do we have additional questions from the audience?